Executive summary. Glutamine is a common amino acid in the body, but current evidence does not show that glutamine itself causes or treats glaucoma. Instead, glutamine is part of the normal glutamate-glutamine cycle in the nervous system, including the retina. In glaucoma, a disease where retinal ganglion cells and the optic nerve degenerate, researchers have wondered whether excitotoxic damage from too much glutamate may play a role. Since glutamine is the main precursor for glutamate, it is studied as an indirect marker of this process. Some experimental studies, mostly in animals or lab models, show changes in glutamine handling by retinal glial cells when pressure or blood flow is disturbed. A few small human studies found glaucoma patients had slightly higher glutamine in the eye's fluids, while others found no difference. Overall, the human data are limited and inconsistent. Glutamine supplements have not been shown to help glaucoma, and no clinical trial has tested this. There is also no evidence that taking or avoiding glutamine changes eye pressure or disease. In practical terms, the main proven treatment for glaucoma remains lowering eye pressure with drops, laser, or surgery, not dietary changes. What is glutamine? Glutamine GLN is one of the body's most abundant free amino acids. It serves many roles: a building block for proteins, a fuel for immune and gut cells, and a carrier of nitrogen between tissues. Under stress or illness, cells use glutamine quickly and it can become conditionally essential, meaning we may need more from food or supplements. Glutamate, GLU, is a closely related amino acid that acts as a major excitatory neurotransmitter in the brain and retina. In contrast, glutamine itself is not an excitatory neurotransmitter. Instead, it is a converter or storage form. Neurons use glutamine mostly to resynthesize glutamate. High extracellular glutamate can be toxic to neurons, a process called excitotoxicity, but glutamine is not toxic and does not directly activate glutamate receptors. The glutamate-glutamine cycle. In the retina and brain, neurons and glial cells recycle glutamate and glutamine in a tight loop. For example, a neuron, such as a retinal ganglion cell, releases glutamate at its synapse. Nearby Miller glial cells, the main support cells in the retina, quickly take up this glutamate and convert it into glutamine. The Miller cell then releases glutamine back to neurons. Neurons take up glutamine and convert it back into glutamate for future signaling. In effect, glutamine is a safe way to mop up excess glutamate. It keeps the fast-acting glutamate neurotransmitter within neurons and prevents glutamate from lingering too long outside cells, which could be harmful. The cycle is illustrated conceptually below. Neuron releases glutamate, glial cell converts glutamate, glutamine, glial cell sends glutamine back, neuron converts glutamine back to glutamate. This recycling ensures that neurotransmitter levels remain balanced. Importantly, disturbances in this cycle, for example, if glial cells fail to clear glutamate, can allow glutamate buildup and potentially cause excitotoxic damage to neurons. Why could glutamine matter in glaucoma? Glaucoma basics. Glaucoma is a group of eye diseases leading to optic nerve damage and vision loss, usually by death retinal ganglion cells, RGCs. The most common form is primary open-angle glaucoma, often associated with elevated intraocular pressure, IOP. Another form is normal tension glaucoma, where nerve damage occurs at normal pressures. Regardless of pressure, glaucoma involves progressive RGC loss. The National Eye Institute and others describe glaucoma as an optic neuropathy, nerve disease, that leads to peripheral vision loss and eventual blindness if untreated. Excitotoxicity hypothesis. Because glutamate is known to kill retinal neurons in lab studies, for example, injecting glutamate into the eye causes RGC death, scientists have long hypothesized that elevated glutamate could contribute to glaucoma damage. Some early studies reported higher vitreous eye fluid glutamate in glaucomatous eyes, suggesting an excitotoxic mechanism. In one review, it was noted that glaucoma patients had about 27 minomens glutamate in vitreous versus 11 minomens in controls, enough to harm RGCs. However, other studies, including Honkanen et al. 2003, found no significant increase in ocular glutamate or glutamine in glaucoma patients. The role of glutamate excitotoxicity in human glaucoma remains unproven. Glutamine's indirect role. Because glutamine is the precursor and breakdown product of glutamate, it is studied indirectly. If glutamate were accumulating, one might see changes in glutamine too. For example, one recent hypothesis is that in glaucoma, Miller glial cells may raise glutamine production in order to keep free glutamate levels low and protect neurons. In effect, more glutamine in eye fluids might reflect an attempt to buffer glutamate. This is only speculative. The Frontiers study, Lillo et al., mentions that higher aqueous glutamine in glaucoma could be a means of keeping the concentration of glutamate under control, thus avoiding neuron death. But whether this happens or matters in patients is unknown. Miller cell and astrocyte changes. Glial cells, Miller cells in retina, astrocytes in optic nerve head, normally regulate glutamate-glutamine recycling. In animal glaucoma models, these glial cells sometimes become reactive or dysfunctional. For instance, experimental glaucoma in monkeys led to higher glutamine labeling in Miller cells, suggesting they were still converting extra glutamate to glutamine. In rat studies, raising intraocular pressure briefly actually blocked the increase in glial glutamine synthetase, GS, that would normally follow glutamate exposure. Only after 1 week of continued pressure did Miller cells resume raising GS as before. This hints that acute pressure spikes might temporarily impair glial glutamate clearance. Such mechanistic findings show that the glutamate-glutamine cycle can be altered by glaucoma-like conditions, but they do not prove that glutamine itself is toxic or protective. They simply underscore that late-stage RGC death in glaucoma could involve metabolic stress in glial cells. Human research. Glutamine-glutamate levels in glaucoma studies in humans have looked for differences in glutamine or related metabolites in the eye or blood of glaucoma patients. The results are mixed and generally not definitive. Aqueous humor eye fluid studies. New metabolomics analyses of aqueous humor, the fluid in the front of the eye, found that glaucoma patients had higher glutamine levels than controls. For example, a 2022 Frontiers in Medicine study reported median glutamine 697 millimommers in glaucoma patients versus 563 millimommers in cataract controls. This was statistically significant, and the authors noted glutamine, but not glutamate, was elevated in treated glaucoma. They suggested this might help keep glutamate low in the eye. However, older analyses of aqueous humor and vitreous have not consistently confirmed this. A systematic review of glaucoma metabolomics noted that some studies found glutamine increased, e.g. Buset et al. 2019, Tang et al. 2021, while others saw it decreased or unchanged, e.g. Meyer et al. 2020. In meta-analysis of multiple aqueous humor studies in open-angle glaucoma, glutamine was often reported as an affected metabolite, but the findings went in opposite directions in different studies. Overall, aqueous humor data suggest there are metabolic changes in glaucoma, but the specific role of glutamine is uncertain. Vitreous humor eye gel studies. Vitreous samples from glaucoma eyes have been measured in a few small studies. Honkanen et al. 2003 measured 16 amino acids, including glutamate and glutamine, in vitreous from glaucoma patients undergoing vitrectomy, usually for other eye problems, versus controls. They found no significant difference in glutamine and no significant difference in glutamate between groups. The average glutamine was 1200 millimommers in both glaucoma and control eyes, with p99. This argues against a large buildup of glutamate or its precursor glutamine in human glaucoma vitreous. Earlier, Dreyer 1996 had reported higher glutamate in vitreous of glaucoma patients, but that finding was not replicated by Honkanen. In experimental eyes, a rabbit model of optic nerve ischemia simulating glaucoma also showed no change in vitreous glutamine, even though glutamate tripled, so human vitreous data to date do not support a glutamine difference. Blood serum studies. There is little data on glutamine in the blood of glaucoma patients. Metabolomics studies of patient plasma have identified many molecules altered in glaucoma, but glutamine specifically has not emerged as a clear marker in blood. For example, Tang et al. 2021 profiled plasma metabolites in POEG versus cataract controls and found some energy-related changes, like purine metabolism shifts, but glutamine was not highlighted as a key finding in their plasma results. The systematic review notes glutamine-glutamate pathways appear among altered pathways in POEG retinal tissue, but evidence from blood is limited. In short, There is no strong evidence that glaucoma patients have abnormal glutamine levels in routine blood tests. Differentiating types of glaucoma. Most human studies so far have focused on primary open-angle glaucoma, POAG, sometimes mixing in normal tension or other subtypes. Aqueous humor studies like Tang 2021 explicitly looked at POAG. Vitreous studies usually involved high-pressure glaucoma, often secondary to surgery. There are virtually no data on glutamine in normal tension glaucoma or angle closure glaucoma specifically. Thus, any small differences found cannot be attributed to one type over another. Animal and lab research, laboratory studies in animals or isolated tissues have explored how glaucoma-like conditions affect glutamate-glutamine metabolism. Key findings include glutamate excitotoxicity in the retina. Basic neuroscience experiments show that applying extra glutamate can kill retinal ganglion cells. For example, injecting glutamate into a mouse eye destroyed the inner retina, including RGCs. This establishes that high glutamate can be toxic, excitotoxic, to retinal neurons. It does not prove it happens in human glaucoma, but it provides a model. Optic nerve ischemia models. In rabbits, delivering endothelin-1, a blood vessel constrictor, to the optic nerve caused ischemia and RGC death. This model of glaucoma resulted in 2.6-fold higher vitreous glutamate with associated cell loss. But no change in glutamine, levels remain 330 mg. This suggests that during optic nerve injury, glutamate can rise abnormally even when glutamine stays stable. Glial cell changes. In various glaucoma models, Miller glial and optic nerve head astrocytes show metabolic stress. For instance, in an acute rat ocular hypertension model, a brief IOP rise caused Miller cells to lose their normal upregulation of glutamine synthetase, GS, that would follow a glutamate surge. After 1 week of sustained IOP elevation, the GS response returned. This implies that short-term pressure spikes may hamper the glial clearance of glutamate. In a monkey glaucoma model, researchers found 25 to 48% higher glutamine immunoreactivity in Miller cells than in normal eyes, indicating that these glia were converting more glutamate into glutamine. In other words, even though RGCs were dying, Miller cells were still actively processing glutamate. These animal studies show that glial glutamine production is altered by glaucoma-like stress, but they stop short of showing glutamine itself is causal. Glutamine synthetase, GS experiments. Glutamine synthetase is the enzyme in glial cells that converts glutamate to glutamine. Experiments in retinal tissue have directly tested its role. Gorovitz et al., 1997. Used cultured chick retinas and showed that increasing GS activity by hormone treatment strongly protected neurons from dying after injury, whereas inhibiting GS with methionine sulfoxamine led to much more death. In plain terms, boosting the glial conversion of glutamate to glutamine saved neurons, whereas blocking it made injury worse. This supports the idea that glutamine synthase and the glutamate-glutamine cycle defends neurons in retinal injury. Other studies in brain and retina have reached similar conclusions. Glial GS helps prevent excitotoxic damage. Metabolomics of retinal tissue. Metabolic profiling of whole retina in animal glaucoma models has implicated glutamate-glutamine pathways. For example, one study found that pathways related to D-glutamine and D-glutamate metabolism were among those altered in glaucomatous retina. These findings suggest mitochondrial and amino acid metabolism in retina glial neurons is disturbed by glaucoma. However, metabolomics of tissues is complex and can't pin a single molecule as the cause. Summary of lab findings. In short, laboratory research confirms that glutamate excitotoxicity is real and that the glial enzyme glutamine synthetase can protect retinal neurons. Animal glaucoma models often show elevation of glutamate and sometimes glutamine changes under stress. Glial cells, Miller and astrocytes, in these models alter their glutamine metabolism. Taken together, these mechanistic studies emphasize that disturbances in glutamate-glutamine metabolism can contribute to retinal damage, but they do not prove that giving extra glutamine or changing diet will affect glaucoma. They simply underline that healthy glial metabolism with adequate glutamine processing is part of normal retinal function. Is oral glutamine supplementation relevant? Absorption and retinal effect. L-glutamine supplements, sold for gut health or athletic recovery, raise glutamine levels in the blood. However, the eye is protected by the blood-retinal barrier and has its own tightly regulated metabolism. There is no evidence that taking oral glutamine significantly changes glutamine or glutamate levels in the eye. Sterile eye fluids and retina rely mainly on local synthesis and recycling of glutamine glutamate. In other words, eating more glutamine probably does not flood the retina or directly alter the glutamate glutamine cycle in any clear way. Clinical trials and case reports. We found no clinical trials or case reports linking glutamine supplements to glaucoma outcomes. No study has tested glutamine for glaucoma. Similarly, no safety alerts specifically mention glaucoma in relation to glutamine. This simply means that A, nobody has proven that glutamine supplements help glaucoma patients, and B, nobody has documented a glaucoma patient being harmed by taking a bit of L-glutamine. What the evidence does not show. Importantly, because there is no research on this specific question, we cannot draw strong conclusions. There is no basis to recommend glutamine for glaucoma. No evidence it improves eye pressure, vision, or retinal health in glaucoma. Likewise, there is no convincing evidence that supplemental glutamine would worsen glaucoma. The idea that more glutamine might fuel more glutamate in the retina is speculative. Normal bodies prevent harmful levels of glutamate through the glial cycle and enzyme regulation. Safety considerations. Glutamine supplements are generally considered safe for most healthy people. Common side effects at high doses can include mild gastrointestinal symptoms like bloating or discomfort. Some special cautions, not specific to glaucoma, are liver-kidney disease. Because glutamine metabolizes to ammonia, very high doses can potentially raise ammonia levels, which could be an issue in patients with severe liver problems or urea cycle disorders. Cancer. Some cancer cells use glutamine as fuel. In active, untreated cancer, doctors sometimes advise caution with high-dose glutamine supplements, although routine diet levels are normal. The evidence is mixed, and glutamine is sometimes given to cancer patients to protect healthy cells from chemo. Because glaucoma patients are usually elderly, one should use judgment if there is active cancer. Neurological conditions. In theory, people with epilepsy or certain psychiatric conditions might be sensitive to any changes in glutamate metabolism. However, there is no specific data on glutamine supplements triggering seizures or mood issues in glaucoma patients. If one has a severe neurological condition, it's wise to discuss any supplement with a doctor. Medications. There are no known dangerous drug interactions with standard glaucoma medications in the context of taking glutamine pills. Still, if you're on medications for any condition, it's always best to ask your doctor before starting a new supplement. Bottom line on supplements. At present, taking L-glutamine is not indicated for glaucoma. It likely will have no benefit for your eye condition. It also is unlikely to cause harm at moderate doses, e.g. a few grams per day, except in the above special situations. But again, these points are general supplement guidance, not specific glaucoma advice. Because to date, no study has tested glutamine in glaucoma. Could glutamine be a biomarker for glaucoma? Researchers have explored whether glutamine or related metabolites in eye fluids could serve as a diagnostic or prognostic marker. Some findings include: the Targeted Metabolomic Study in Frontiers in Medicine 222 identified glutamine as one of 11 compounds significantly different in aqueous humor of glaucoma patients. They even calculated a cutoff value for glutamine to distinguish patients from controls. However, the authors noted that many other compounds, like certain lipids and kynurenine, as well as glutamine, were altered, and they did not claim glutamine alone is diagnostic. Recovery of a single reliable biomarker for complex diseases like glaucoma is challenging. A review of multiple studies summarized that glutamine often appears among altered metabolites in glaucoma aqueous humor, but different groups reported opposite changes. This inconsistency suggests glutamine is not yet a clinically useful standalone marker. No standard clinical test measures glutamine in the eye. We only measure eye pressure and do imaging. A biomarker would ideally be something easily measured in blood or urine. Glutamine has not emerged as a simple blood or urine marker for glaucoma risk or progression. In summary, glutamine and related amino acids are being studied in the lab for glaucoma, but there is no validated glutamine test for glaucoma in the clinic. If a future panel of metabolites, including glutamine, were proven to predict glaucoma risk or progression, that would need much more research and regulatory approval. Right now, it remains an interesting research question, not a tool for patients. What we know, glutamine is an amino acid, not an excitatory neurotransmitter. It is used by glial cells to safely shuttle ammonia and nitrogen, and neurons convert it to glutamate for signaling. Glaucoma involves retinal ganglion cell, neuron, death, and optic nerve damage. The main known risk factors are high eye pressure and age-related changes. Excess glutamate has been proposed as one damaging factor, but human evidence is unclear. In glaucoma patients studied so far, ocular glutamine levels show at most subtle changes. Some eye fluid studies report slightly higher glutamine in glaucoma, while others find no difference. Blood glutamine in glaucoma has not surfaced as a clear signal. In other words, patients with glaucoma do not consistently have abnormal glutamine levels in the eye or blood compared to others. Animal lab models support that glial glutamine handling can change under stress. Experiments show that Miller glial cells alter their glutamine synthetase activity when pressure or blood flow to the eye changes. Increasing glial GS, hence making more glutamine, protects retinal neurons in lab injury models. These findings indicate that a healthy glutamate-glutamine cycle is important for retinal neuron survival. Glutamine supplements have no proven effect on glaucoma. There are no clinical data suggesting benefit or harm. Most evidence about glutamine comes from general nutrition or critical care studies, which show glutamine is generally safe for most people, but is not a specific treatment for eye disease. What remains uncertain? Causality versus correlation. Even if some studies find higher glutamine in glaucoma eyes, it's unclear if that is a cause or effect. It may simply reflect glial cells working harder to cope with cell injury. We don't know if altering glutamine levels would change disease course. Exact triggers of RGC death. Glutamate excitotoxicity is a plausible mechanism in glaucoma, but definitive proof in humans is lacking. Other pathways, like blood flow, inflammation, mitochondrial dysfunction, play roles. The contribution of glutamate-glutamine imbalance is still under investigation. Variations by glaucoma type. We do not have detailed data on glutamine for different glaucoma subtypes, e.g. normal tension, angle closure, pseudoexfoliation. It's possible metabolic changes differ among types, but studies have mostly focused on open-angle glaucoma. Biomarker validity. Can glutamine, or a panel including glutamine, reliably predict glaucoma onset or progression? This is still hypothetical. Small metabolomic studies hint at metabolic signatures, but they need replication in larger multicenter trials. Currently, no clinical test exists. Long-term supplement effects. Almost nothing is known about very long-term use of glutamine in patients with chronic eye diseases. It is uncertain whether modest excess intake could subtly affect neuronal health over many years. No research has addressed this. Supplement safety. What can and cannot be concluded? Can be concluded. L-glutamine supplements are generally safe for healthy adults at usual doses, 10 to 20 grams per day. They are even used in critical illness without major safety issues. If someone with glaucoma takes a low-dose glutamine supplement, e.g. 5 grams once daily, they are unlikely to experience eye-related problems from it. In fact, glutamine is GRAS, generally recognized as safe for normal use. Cannot be concluded. We cannot conclude that glutamine supplements will protect the optic nerve or improve vision in glaucoma. No evidence for that exists. We also cannot conclude that glutamine supplementation will harm glaucoma by increasing glutamate in the eye, because again, this has not been studied. In short, the effects of added glutamine on glaucoma are unknown. Bottom line, if a patient with glaucoma asks whether they should take glutamine, the honest answer is that we have no data to support it. It's not part of standard glaucoma care. On the other hand, for most people, glutamine is not dangerous, but it's also not expected to fix glaucoma. Patients should therefore focus on treatments that are proven, eye drops, lasers, surgeries to lower pressure, and discuss any supplements with their doctor. Conclusion for patients. To put it simply, glutamine is not currently a factor that patients need to worry about in glaucoma. It's a normal amino acid used by cells in the eye, among many other places. While scientists have studied it as part of the glutamate recycling system, There is no clear evidence that glutamine itself causes glaucoma, nor that taking extra glutamine will make glaucoma better or worse. The medical literature so far suggests only subtle or inconsistent changes in glutamine levels in people with glaucoma. For you as a patient, the most important point is this: treat glaucoma by controlling your eye pressure and following your ophthalmologist's advice. That remains the gold standard approach. There is no proven benefit to adding a glutamine supplement for glaucoma, and specialists do not currently recommend it. If you happen to take glutamine for another reason, for example, gut health or general wellness, you can continue under your doctor's guidance, but don't expect it to influence your glaucoma. Always let your doctor know about any supplements, especially if you have liver, kidney, or cancer conditions, so they can advise on safety. In summary, the science does not support a direct link between glutamine intake and glaucoma progression. Keeping your intraocular pressure low with prescribed treatments is still the key to protecting your vision. If you have more questions about diet or supplements, bring them up in your eye care appointments. Your doctor can help you understand what is evidence-based. Table: key studies on glutamine in glaucoma, study year, sample model, glaucoma type, metabolite, measured, main finding, evidence, strength. Human vitreous, mixed undergoing vitrectomy, vitreous glutamine, 12 et al. 2003. Human vitreous, N=8, glaucoma, 17 controls, eyes undergoing vitrectomy. Glutamine and glutamate. Vitreous glutamine, twice 1200 mg in both groups, no difference. Glutamate likewise unchanged. Clinical, moderate, small sample. Lillo et al., 2022, human aqueous humor, glaucoma versus cataract, primarily open angle, well controlled with meds. Glutamine, other metabolites. Glutamine was significantly higher in glaucoma AH, median 697 versus 563 mg. Glutamate unchanged. Clinical, moderate. Kim et al., 2000, rabbit model, optic nerve ischemia via ET1 pump. Experimental ischemic glaucoma model. Glutamate, glutamine, vitreous. Vitreous glutamate rose 2.6 times in ischemic eyes, p.4, but glutamine stayed virtually identical. Animal, moderate, Shen et al. 4. Rat retina episcleral vein cauterization IOP model, experimental ocular hypertension, glutamine synthetase in Miller cells, short-term IOP elevation alone did not change GS levels. Injecting glutamate increased GS by 40%. Acute IOP blocked that response. 1 week IOP restored it. Animal mechanistic. Osborne et al., 1998. Monkey retina, experimental high-tension glaucoma, experimental glaucoma glutamine, immunolabel in Miller cells. Glutamine immunoreactivity in Miller cells was 25 to 48% higher in glaucomatous eyes versus controls. Miller cell numbers were unchanged. Animal, moderate. Gorovitz et al., 1997. Cultured chick retinal tissue, in vitro injury, retinal injury model, glutamate toxicity. Glutamine synthetase, GS activity, inducing glial GS expression, protected neurons from degeneration, inhibiting GS with MSO, greatly increased cell death. Lab, mechanistic. What we know: glutamine and glutamate level studies. Glutamine is a non-toxic precursor to glutamate, abundant in cells. Glaucoma damages RGCs and optic nerve. High glutamate can kill RGCs in lab models, but human glaucoma glutamate data are mixed. In glaucoma patients, eye fluid glutamine shows minimal or variable changes, as above. Animal studies highlight glial GS as protective. Boosting GS, more glutamine formation, helps injured retina, while blocking it hurts neurons. Glial cells in glaucoma models often make more glutamine, converting excess glutamate. What remains uncertain: whether glutamate excitotoxicity actually occurs in glaucoma patients. Whether observed glutamine changes are cause or effect of damage. If glutamine, or related metabolites, could reliably signal early glaucoma risk or progression, current studies disagree. The impact of dietary glutamine on ocular glutamate-glutamine balance is unknown, no data. Supplement safety. Oral glutamine is generally safe for healthy adults. No evidence it improves or worsens glaucoma. No known interaction with glaucoma meds. Caution in liver disease, ammonia, advanced kidney disease, or active cancer. Some tumors use glutamine, but these are general issues, not glaucoma specific. Patients with neurological disorders or on multiple drugs should consult a doctor before taking any supplement. What we know: glutamine versus glutamate. Glutamine is not the same as glutamate. Glutamate is an excitatory neurotransmitter, whereas glutamine is its precursor and a safer transport form. Glutamine does not itself excite neurons or cause toxicity. It circulates between neurons and glial cells to regulate glutamate levels. Glaucoma basics. Glaucoma involves progressive loss of retinal ganglion cells and optic nerve damage. It is primarily managed by lowering intraocular pressure. Proposed additional injury mechanisms include reduced blood flow and possible glutamate excitotoxicity, but definitive proof in humans is lacking. Human studies measuring eye fluids in glaucoma patients generally show no large glutamine abnormalities. Some show slightly higher glutamine in glaucoma eyes, others show none. Overall, glutamine levels are at best a subtle signal and not a consistent marker of disease. Animal and lab studies. Lab models confirm that excess glutamate can kill retinal cells and that glial glutamine synthetase is protective. In animal glaucoma models, Miller cells often increase glutamine production, likely to buffer glutamate. These findings indicate that maintaining glutamate-glutamine balance is important for neuron survival, but they do not demonstrate that altering glutamine by diet or drugs will treat glaucoma. Supplements. There is no proven benefit of taking glutamine supplements for glaucoma. Likewise, there's no evidence they worsen glaucoma. In general nutrition, glutamine is safe for most people. The only glaucoma treatment proven to slow vision loss is pressure control, not dietary changes. What remains uncertain: causation. We do not know if glutamate excitotoxicity actually happens in most glaucoma cases. Human evidence is mixed. If it does occur, glutamine's role as precursor or buffer is still hypothetical. Biomarkers. It is uncertain whether measuring glutamine or related metabolites in eye fluid could ever reliably diagnose or track glaucoma. Current studies disagree on whether glutamine levels rise or fall in glaucoma. More research is needed. Supplement effects. No studies have tested whether oral glutamine alters glaucoma risk or severity. Thus, any effect, good or bad, of dietary glutamine on the retina is unknown. We only have indirect evidence from basic science. Long-term outcomes. We don't know if very long-term, subtle increases or decreases in glutamine could influence glaucoma. Existing human studies are cross-sectional, single-time point, and animal lab studies are short-term. Supplement safety. What can and cannot be concluded. What we can say: glutamine supplements, typical doses of a few grams per day, are generally considered safe for most people. If your glaucoma treatment plan is optimal, pressure well controlled with drops, laser surgery, taking glutamine by mouth is unlikely to have a dramatic effect on your eyes one way or the other. What we can't say: there are no studies showing that glutamine supplements improve vision, nerve health, or eye pressure in glaucoma. There are also no reports of supplements worsening glaucoma specifically. In short, any direct impact of glutamine pills on glaucoma is unknown. General cautions, not glaucoma specific. If you have severe liver or kidney disease, extra glutamine could raise ammonia levels and adversely affect you. Some cancers preferentially consume glutamine, so doctors sometimes advise caution in patients with active tumors. These issues are about general health, not about glaucoma. Bottom line: for a glaucoma patient, taking normal doses of glutamine is probably harmless but not helpful. It's not a substitute for doctor-prescribed treatments. Always inform your doctor about supplements so they can advise on your overall health context. Final answer: plain language. To put it simply, glutamine is not the key to glaucoma. Glutamine is a harmless amino acid that participates in normal nerve cell metabolism, but there is no strong evidence that it causes glaucoma, or that taking more or less of it will change your glaucoma. Scientists have studied it mainly because of its link to glutamate, a nerve transmitter that can damage eye cells in lab experiments. Some early lab and animal studies suggest the cells that support the retina might make more glutamine in glaucoma, possibly to protect the nerve cells. But in real patients, the differences in glutamine levels have been small and inconsistent. Importantly, no clinical trials have tested glutamine supplements for glaucoma. That means doctors have not found any proof that eating glutamine-rich foods or taking glutamine pills helps vision or slows glaucoma. In fact, eye specialists do not recommend any special diet or supplement, including glutamine, for glaucoma because it's not shown to work. The best way to manage glaucoma remains the treatments your eye doctor prescribes, like pressure-lowering eye drops or surgery. If you are a glaucoma patient wondering about supplements, there's no evidence to start or stop glutamine specifically for your eyes. Taking a normal glutamine supplement is unlikely to make your glaucoma worse, but it also is not likely to make it better. Always check with your doctor before adding any new supplement, especially if you have other conditions like liver, kidney, or cancer issues, or are on cancer medications. In summary, focus on proven therapies, control your eye pressure, and follow your eye doctor's plan. Glutamine is simply not a proven player in the story of glaucoma. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.